Bismillahirrahmanirrahim. I am really thankful to the organizing committee of 7th International Conference on Applied Zoology and in collaboration with the University of Sargodha for providing me this opportunity to share my thoughts on this esteemed forum. I am Dr. Sabah Farooq from the Department of Biology, Allama Iqbal Open University, and today I am going to discuss evaluation of frequency of Acinetobacter from surgical site infections and assessment of multiple antibiotic resistance index. The presentation includes introduction, objective of the study, methodology, results, and conclusion. Acinetobacter baumannii is a gram-negative organism which is widely distributed in environment. It is found in soil and water as well. It is an opportunistic pathogen in human which is responsible for nosocomial infections. This organism is rapidly acquiring multi-drug resistance. Multi-drug resistance is the attribute by which bacteria can resist one antibiotic belonging to three different antibiotic groups. And this multi-drug resistance is an emerging threat for therapeutic and healthcare facilities. Nosocomial infections are responsible for hospital-acquired pneumonia, septicemia, meningitis, urinary tract, and wound infections. Objectives of study include isolation and characterization of Acinetobacter baumannii from surgical site infections, identification of bacteria using biochemical and automated profile indexing, profiling of resistance of antibiotics against different antibiotic groups. For this purpose, 165 surgical site infection samples were collected from different healthcare facilities of Islamabad. These samples were subjected to standard microbiological procedures for bacterial isolation. 33 bacterial isolates were obtained, which were subjected to morphological and colony examination, followed by microscopic examination. Based upon gram staining and biochemical characterization, including catalase test, indole test, urease, motility, triple sugar iron utilization test, citrate utilization, oxidase test, 9 bacterial isolates were considered as base. This slide represents the isolation of Acinetobacter baumannii from different surgical site infection samples. Most frequent isolation was from catheter tip, followed by suction pump. Only one isolate was obtained from blood and one isolate was obtained from pus swabs. The growth pattern representative of Acinetobacter baumannii cultivation are shown on blood agar plate and McConkey agar plate. Biochemical tests carried out for the identification of organism include catalase test, indole test, citrate test, urease test, oxidase test, motility test. All Acinetobacter baumannii isolates were positive for catalase test, negative for indole test, positive for citrate utilization test, negative for urease, negative for oxidase test, and negative for motility test. Further, species level identification of Acinetobacter baumannii was carried out by using API strips, automated profile indexing, and recording all the results of these tests. Identified isolates of Acinetobacter baumannii were then used for antibiotic susceptibility and resistance profiling by Kirby-Pure method. In this process, test diffusion method is used to assess the antibiotic resistance of the test organism by using the zone of inhibition along with the comparison of CLSI standards. The bacteria were classified into resistant, intermediate resistant, or sensitive to the selected antibiotic. After recording the resistance and sensitivity pattern of bacteria against different multiple antibiotics used, MAR index was calculated for all the 9 bacterial isolates. Isolate number 1 was resistant to 9 out of 11 antibiotics used, exhibiting 0.81 MAR index. Second isolate was resistant to 9 out of 10 antibiotics used. The third exhibited resistance to 10 out of 11 antibiotics used. Both gave MAR index of 0.90. Fourth isolate was resistant to 8 out of 11 antibiotics used. Fifth isolate was resistant to 9 out of 11 antibiotics used. Only one isolate gave resistance to all antibiotics used against it, exhibiting MAR index of 1, which is extremely high. Seventh isolate was resistant to 2 antibiotics out of 7 used, and it exhibited the least MAR index. Eighth isolate exhibited 10 out of 11, and 9 also 10 out of 11 resistance to different antibiotics used. Keeping in view the results of study, we can say most Acinetobacter baumannii isolates had MAR index greater than 0.2. This index represents the emergence of these isolates from a high-risk contaminated niche where antibiotics have been extensively used. The isolate, which was resistant to all the antimicrobial agents used against it, was isolated from a catheter tip. It is now the need of hour to regulate the use of antibiotics and to stop the spread of antibiotic resistance across different bacterial genera so that the burden of hospitalization and extensive antibiotic use can be minimized. With the take-home message of regulation of antibiotics at our individual level and national level, I will conclude my presentation. That is all from my side. Thank you very much for listening. Thank you very much for your attention.